Well, hello, fellow Rosarians. As you know, we have a monthly meeting, and 2 months ago, we had a really good chat with Dr. Mark Windham, and he spoke with us about Rose Rosette Disease. And at the time, I did not record it, and I've been kicking myself since. And fortunately, I've been able to convince Dr. Mark to come back and give us another little chat. So thank you so much for joining us, Dr. Mark.
Happy to do it.
All right, so I'm going to pass it over to you if you want to share your desktop.
All right, today we're going to be talking about Rose Rosette, some of the advances and updates that we have for this disease. And during this presentation, I'm going to be answering questions that was sent to me by the Rose Geeks. And so I'll have the questions listed on the slides as well. And so we know what Rose Rosette looks like, but we've also learned that that's not good enough. What we really need to do is to understand that there can be as many as 80 times more mites on symptomatic tissue than asymptomatic tissue. And so this is incredibly important. What this means is that if you leave a rose in your garden that has Rose Rosette, symptoms that it's going to be spewing, ballooning mites that are carrying the virus to your other roses. It also tells us that if for some reason you can't remove the rose at that particular moment, maybe you're going out of town, maybe you've hurt your back and you just can't do it right at the moment. Well, if you can't, at least cut out that rosette and get rid of it, because that is where the bulk of your mites are going to be located, is on that rosette. So you got to get rid of it, but the rest of the plant is infected. You can't cure it. Practically all the mites are on that symptomatic tissue. And so what we want to do is to know when we should take action. And to take action is when you can first detect symptoms to Rose Rosette. And this is when we have one symptomatic shoot. Usually the stem is thickened. The thorns on the stem, there may be more than what there should be. The thorns can be larger. Sometimes they're large and sort of elastic. You can bend them. The leaves are thin. They're said to be strapped like a razor strap, which is long and thin. The flower buds can be distorted as well. And you want to catch it at that stage. You don't want to catch it at the rosette stage, which is in the picture on the right. So, because by then you've already got mites spreading to your other roses. So you have to be vigilant and you have to look for those early symptoms. Now, I mentioned the mite. The mite that transmits Rose Rosette is very, very tiny. It's so much smaller than a spider mite. The difference between an eriophyid mite and a spider mite, which we can see, not well, but we can with a magnifying glass. You can't see an eriophyid mite with a magnifying glass. It's about the same size between a bunny rabbit and an elephant as an eriophyid mite is to a spider mite. So they're real small. They're going to peak in late summer to early fall. And usually, at least in Tennessee, up until early June, the populations haven't grown at all from the winter. But once they start and once it warms up, a female mite can produce about one egg a day for 30 days. And so she will, in a course of a month, she will give birth to 30 eggs. And those eggs will hatch in 1 to 2 days. And by 5 days later, those first ones are producing eggs of their own. And so this is why the thing can just seem to explode. Eriophyid mites do not have legs, they don't have eyes, and they don't have wings. We don't think that they can steer themselves to a rose, but it's possible that they might be able to orient themselves, but they really can't see, so we don't know what that really buys them. They're niche mites. They get down in nooks and crannies, and they're hard to get at with pesticides. And the picture on the right is a colony of eriophyid mites. The mites are conical shaped, and those round spears that you see are eggs. And so if you peel it back and you look down in there with a scanning electron microscope, this is what you're going to see. Now, we're going to watch a short video here, and this is going to allow you to see what these things look like when they're moving around. And I'm going to skip this out till about 40 seconds. And there are the eriophyid mites. And you can, and those globule looking things, those are rose hairs on a leaf. All that stuff there are different types of hairs and glands that are on a rose leaf. And this is how they disseminate themselves. They can, even though all four legs are at the head end of the mite, they can stand up on their tail end and they secrete a little drop of glue that holds them in place, and their legs will act like sails as they're moving them back. And when they catch the wind, they will release another chemical that will dissolve that little bit of glue, and they pop off the leaf like a cork out of a pop gun. And so, and here is another mite just crawling around, that when they find a place they want to feed, they have 5 stylots in a bundle that they will pierce the leaf cell with, and then they'll secrete through those stylots, think of them as soda straws, some digestive enzymes, and then they'll suck the partially digested cell contents back into them. Unfortunately, when they put those digestive enzymes into the cell, they can put virus particles into that cell as well. This is a non-lethal wound, and that's why they're such efficient vectors. Now, mites ballooning in the wind is how it gets moved around in your garden. But mites and the virus can show up anywhere. And that's because roses are moved in interstate shipment, and infected plants with phyliferous mites can move from A to B or B to A or B to C or D very, very rapidly. And so we have this issue that just seems to pop up out of nowhere. And this has been going on for a number of years. And because of that, it's pretty widespread now. And in this cartoon, this is what happens on a windy day. You know, if you have a neighbor down the road, or there is a strip mall that has a bunch of shrub roses in front of the strip mall, but they're not taken real good care of, and some of them are symptomatic for the virus, and the mites are ballooning off, they can just drift right into your yard, right in onto your roses. As it shows here, look, some drifters coming into town. That's exactly what can happen, but this is considered short. When I say short distance spread, we're talking hundreds of yards to a couple of miles, okay? Where long distance spread would be like from Ohio to California, or from California to Florida. Now, some of the questions that have been asked is this spread throughout the US? And do certain zones have higher susceptibility compared to others? Well, this is the map from January 2nd, and it would not have changed much because the mites are not active at this time of year. And neither are the roses in most places, except for the deep South, with roses really, down in Florida, they're holding rose shows right now, where it's going to be 23 degrees this Saturday in Knoxville, where I'm at. But anyway, you can see every one of the counties that's in green has been reported to have Rose Rosette. Now, Rose Rosette was first in a few western counties, including Wyoming, Idaho, Northern California, and Manitoba, Canada, which isn't on the map, back in the early 1940s. And since then, it has been found in quite a few eastern states, especially. Now, even though every county where I'm at in Tennessee is not green, we assume they all are. That just means it hasn't been reported there. Okay, so this tells us that in the eastern US, it can appear just about anywhere. And so what does Rose Rosette and the virus have to do with distribution in the Deep South? Well, those dots on the map is where a graduate student from my lab, Katie Soto, who's now graduated, and this paper's been published, all those dots represent where she was sampling for both the vector mite, Phylacopsis fructiphilus, and the virus, Rose Rosette virus. And what she found was the red line is where the virus was. You go below the red line and roses look good. You get above it and they're all sick. And the black line below that is how far south did she find the vector? So the vector is much farther south than the rose. But we've had a number of cases of Rose Rosette reported in Florida, both in the Panhandle and in Central Florida, where the virus and the mite were introduced into Florida. And guess what? Mite populations don't survive very well, and the virus never spreads. In fact, that's true for all that area down there. And so this tells us that there's something going on. Now, if we go over to Texas, Texas doesn't behave like these far southeast and deep south states do. And we'll have virus all the way down into Houston. And we don't know what the difference is. There are some theories as to why this happens, but we haven't been able to pin that down because we haven't been able to get funding to look at that. So, I mentioned Florida, I mentioned Houston. You'll notice that there is a spot all the way over there in California that's in a circle. This is where Wesco, California is. And a few years ago, we had an outbreak of Rose Rosette in an area of California where it had never been reported. And about 30 miles away in Bakersfield, California, there was also a report of it on some landscape roses. And this is what happened in weeks. Some roses in 2016, some roses were brought from Ohio to California to Weeks production area and where they do all their breeding. The virus spread throughout their roses, both in their breeding blocks and in their production blocks of roses. They lost millions of dollars and destroyed roses, lost sales and control costs. Look at the size of that rose field. Hiring an airplane to spray miticides could cost $50,000, $70,000, $80,000 for just one application. And they were having to do this about every 10 days to 2 weeks. Weeks was very open about what went on. And they undertook a Herculean effort. They destroyed a tremendous number of roses. But today, Weeks is as clean as anyone. But the message here is that it can show up and it can destroy a lot of roses before you even realize you have a problem if you're not watching for it. Now, another place where this virus can be found is in Multiflora rose, and it can harbor RRD throughout the eastern United States. Multiflora rose is exotic. It's from Europe. It was brought over by some of the early settlers from Europe into North America. It was then in the 1930s, the CCC, Citizens Conservation Corps, they planted Multiflora rose for erosion control. And many states in the East had millions of Multiflora roses planted. And once they're planted, they're very difficult to get rid of because each adult Multiflora rose plant can produce approximately 250,000 seed every year. And those seed can persist on the ground for up to 20 years. So the seed load under Multiflora roses in the wild is extremely high. And it would be in the millions. And it's almost impossible to get rid of. But the problem is, we don't know the extent of this issue. We don't know if Multiflora rose is infected throughout the eastern United States. We know the rose is there, but we don't know if they're infected. We don't know if the incidence of infection in Multiflora rose is higher in some areas than others. These are the kind of questions we need to be asking, but to be honest with you, there's just not funding for it. So one of the questions is, what do we do if we see RRD at a commercial location? For example, a McDonald's or Lowe's or some other box store, or maybe in front of a strip mall. You can go in and talk to the stores there. They don't seem to care. And the comment was, my extension office doesn't seem to care. Well, let me explain to you what's going on. And it's not whether they care or not. It's just that they're sort of hopeless. You see, in this particular case here, this is a big box, some big box stores that are in the Knoxville, Tennessee area. And in both cases, I found Rose Rosette. Now in the bottom picture, that was a bed of roses that were in a large raised container right outside the garden center. And right across a metal fence, but just had rails in it. So mites were going to move back and forth very readily. There were roses for sale, not 15 feet away. The box store turned out that it had no control over those beds. They don't own the store, they lease it. They had no control that there were roses in the bed. They had no control over the fact that there were diseased roses. And so they couldn't do anything about it. They could complain, but that didn't mean that anything was going to be done. Another example of this is it was a hedge in Murfreesboro. And this is at a large strip mall shopping center type place. That hedge, it's hard to get the perspective on it. That hedge is almost 200 feet long. And in 2011, the shrub roses looked great. 2012, many of them had died from Rose Rosette. 2014, well, we're back to uniformity again. They're all dead. Now, I tell you that in 2011, it looked great. You said, Doctor, did it kill them that fast? No, there was Rose Rosette in it. But from a distance, as you're driving by in your car, you didn't notice it, but it was there. And in another year, they had a harsh winter, and the roses just didn't survive because Rose Rosette virus will prevent starch accumulation in the roots. And this is the energy source for roses during the winter. Now, this is another case where I'm at a fast food place, and they had about 50 bushes on the property. All of them were symptomatic for Rose Rosette. We tested them. They were positive for Rose Rosette. And across the street, and when I'm taking this picture, the wind is blowing and it's hitting me in the back. So the wind is blowing across those diseased roses towards that red arrow. And that red arrow is at a very large garden center. And that arrow is where roses are for sale. So there was no question that mites are blowing there. Now that's a very large garden center. They had roses from certified from weeks and from star roses. And here's the problem. So the mites blow from these infected bushes to those. They spread the virus, but it may be 6 months to a year before those plants are symptomatic. But they got the company's tags on it. So when the virus does show up, when symptoms do become apparent, the customer is likely going to say, well, it's a star rose and they were selling plants with virus. No, the virus got on them at the garden center. Okay, but how do you prove that? Who's at fault here? You see, just like at the box store, this fast food restaurant did not own those roses. They told me who the landscaper was because they had seen the trucks. I called and talked to them. They knew the plants were infected. They told the investor group that owned the building that fast food store was at, that investor group was out of state. And it was made up by more than a dozen individuals. I dealt with a similar thing with a hotel where there were 67 different investors. And you got to get them all to agree. You know what the investors told the landscaper? Or the bush is dead? No. Do they still have flowers on them? Yes. So why do you want us to spend $10,000 to pull them out of the ground and dispose of them and then buy something else to put in their place? We're not doing it. So they sit. It's not that the store doesn't care. They don't have any control. The people who do have control don't even live in that state. So this is a very complicated thing. Now, your extension services have investigated this. They found the same thing out that I did. It's not that they don't care, there's nothing they can do in a case like this. Shreveport, Louisiana, when Rose Rosette was first found in the city, it has not been found at the American Rose Gardens at the ARS headquarters, which is outside of Shreveport. But in downtown Shreveport, they found it. And they tried to get an ordinance passed where the city could require diseased roses to be taken out. But the city council didn't want to go along with that. They didn't want more regulations of businesses. So this is a very difficult problem. Prevention. If I remove a rose with Rose Rosette, what's the proper way of disposing of it? Can I plant in the same location? Can I plant in a pot near that location? All good questions that Rose Geeks have asked. So let's take a look at this. We already know that symptomatic plants should be rogued immediately. So you're going to dig the root ball up. I don't know where that F came from. You dig the root ball up and you replant the rose in a hole 1 week later. Okay, so you can go right back in. Why 1 week? Mites can survive off a host up to 5 days. Now, they're not going to hop from the soil back up there, but it's theoretically possible that one might blow off a dirt clod back onto the rose. But in 5 days, they're dead because they can't use a dirt clod for food. So they're gone by 5 days. So you can go right back in the same hole. We used to say that before you dug the rose, that you put a large plastic bag over it. Use some tape to tie it tight around the bottom of the stem and then dig the rose out so that as you're removing it from your rose garden, other roses, the mites couldn't fall off. But we've done research, we published it since then to show that, even if you cut the rose off, the mites don't fall off for 36 to 48 hours. So unless you're going to just leave it laying there for a while, you don't have to bag it, but I wouldn't shake it up and down like shaking a gourd either. I just get it and take it out of the garden, but you don't have to bag it. Now, some of the other questions were, have any growers been affected? Well, we talked about that with weeds. And should we worry about bare root or potted roses from an affected area? Well, whether they're bare root or in a pot with foliage on them doesn't really matter. That's not going to help you one way or the other. It's possible If they have foliage on it, you might see some virus symptoms, but remember, they can become infected and not show symptoms for 6 months to a year. So that's not necessarily going to buy you anything. Can it show up in one of those areas where roses are being produced? Yes, Weeks taught us that. It can show up anywhere. Should you be concerned? You bet you should be concerned. Should you stop buying roses? Of course not. But what this means is that when you get new roses in and they leaf out for that first growing season, you got to watch them, because symptoms could show up at any month during that first growing season. So we want to make sure we're being careful and watching them. Now, here's a case of a wholesale nursery in Georgia. The nursery produced 500,000 container roses. In one block of 40,000 coral drift roses, over 70% of them became symptomatic. Now, that didn't happen overnight. They didn't catch it, because this particular nursery has tens of millions of containers of different plants, and it just got by them. Those roses, though, were worth $10 to the nursery. Okay. Now, that's a wholesale nursery. It's going to go to a retail nursery. By the time it gets to you, it's $20. Okay. But that cost them $400,000. Now, that nursery used to grow 1 million roses, but due to other RRD issues and vests, the next year they only had 200,000. They were down 5-fold. And they were thinking, and they have got out of the rose business altogether, because they couldn't stop the bleeding, because they let it get out. I don't know if there was any Multiflora rose in the woods near the thing. And if they don't own the property, there's nothing they can do. So this can be a tough thing, but it can show up anywhere. And we've mentioned Weeks, so we're going to skip that. Now, we talked about asymptomatic foliage already, but I want to mention this again, because it makes sound epidemiological sense to remove that rosette if you can't remove that bush. So now we're at management. And this is one of the first things we're going to do. We're going to rogue an infected bush. But if your back's hurt, maybe your daughter's getting married and you don't have time to deal with it right now, you got to try to take the 5 minutes to get rid of to bag it and get rid of it. And that would help. And the University of Tennessee has a PDF. You search it, early detection of Rose Rosette with Google. You can download the PDF on the left. You can type in University of Tennessee Rose Diseases, Identification and Management, and you can get that particular thing as well. So, but what if there's RRD in my neighbors? Do I have to give up? Can I never grow roses again? How do I protect my roses from wind-blown infection? And if I'm in a high-risk area, can I use mite or another miticide once a week to help with things? Well, the American Rose Society Research Trust sponsors some research in which we looked at answering some of those questions. And that graph is a summary graph of evaluating fast-growing non-host all grasses or non-host for the virus. And we were trying to see if we could use grasses instead of a fence, which you could use as well. This grass got to be about 7 feet high and grew very dense together and formed a great barrier. And as you can see, did it stop it completely? No. But over a 5-year period, it really slowed down the amount of virus that was able to get into those roses via wind-blown viviparous mites. Viviparous meaning they carry the virus. So barriers are not the end all, but they can certainly help. Whether it's a 6-foot privacy fence, assuming your roses are not 6 feet high, whether it's a privacy fence, a building, all of these types of things can help. As far as miticides go, we have found 4 miticides, and we looked at others that are effective reducing populations of Phyllocoptus fructiphilus, which is the mite vector for Rose Rosette virus, and it protected the roses from getting the virus because it killed the mites. Now, the problem is those 4 miticides, you have to have legally to use it. And I would never recommend trying to get these things, and you could. But if there is an accident or your neighbor's grandchild gets sick and they saw you spray, didn't matter you're spraying a miticide or not, and they say you're the reason why my neighbor my grandchild got sick. If they subpoena your records as to what pesticides you have, and they find a commercial application only pesticide in your home garage, and you don't have a commercial pesticide license, you'll sort of be like a drunk driver in an accident. Whether it's the drunk driver's fault or not, if one driver was drunk, guess what? They're going to be held at fault. And so to protect yourself, you don't want to do that. The good news is that the active ingredient in Tile Star is bifenthrin, and there's over 200 products with bifenthrin in it that can be used for homeowners. And on the right side is 2 of those products that I could have ordered off of Walmart.com. And both of them would be as effective for reducing mite populations as any of those 4 on the other side, but you need a commercial license for it. Only difference is instead of costing $125 a container, they might cost you $14.95. So is it as concentrated? No, but you don't really need it to be enough for one season for sure. But we have another problem. When do you start spraying? How long do you spray? Well, we have data for 2021 yet, but it hasn't been blessed by the statistician yet. So I'll show you what has from 2020. And what we find is there's not a whole lot of movement, in, at least in East Tennessee, and also now we know in Middle Tennessee and West Tennessee. So if you live somewhere else, you just have to sort of guess the weather. But the key is until the time after the first flush of roses, there's probably not any buildup of mites around you in your garden or in other roses around you. But sometime after that first flush of roses is over, and before the second cycle of bloom comes out, the mite populations are going to start to go up. So we tell people, if you don't know when you need to be starting that first spray, it's about the time you're deadheading that first flush of rose blooms off your bushes. And then you're going to have to do it every 2 weeks if you choose to go that route until first killing frost. Because now we know we can extend that chart all the way out to October in Knoxville. And until we have a killing frost, the populations remain pretty static, but relatively high. So we don't suggest, but we have another problem. And that problem is how we know the miticides. The problem is, can we develop resistance? Remember, we only got one for homeowners to use bifenthrin. So right now, I don't even recognize using them. We don't know if you should do it at 2 weeks, 3 weeks, or 4 weeks. We're pretty sure 4 weeks is too long. But we think we may be able to stretch it to only using the miticide every 3 weeks. But we need to collect some more data before I'm confident of that. But that's what it looks like right now. But we don't have enough data for me to be confident. And what does that mean? That would mean 3 weeks versus 2 would mean that we're over a period of a month, we're using 50% less pesticide in the environment. So we have a lot of work to do on this particular area. Some of the other questions: are any varieties more susceptible to RRD? Is Multiflora rootstock more susceptible to RRD? And compared to what? Is Dr. Huey, is it more susceptible? The answer is, yeah, it is. So just about any rootstock is going to be susceptible. Are there specific plants that are carriers more susceptible to the mite that should be avoided in rose gardens? Yeah, roses. You see, Rose Rosette virus and Phyllocopsis fructiphilus only go to members of the genus Rosa. So it only goes to roses. It doesn't go to raspberries. It doesn't go to strawberries. It doesn't go to blackberries. It doesn't go to apples. All of those are in the Rosaceae family. But in the genus Rosa, which is roses, yeah, well, just about anything there is fair game. And we have looked at over 1,000 different roses. We've looked at 28 rose species. We've looked at multiple accessions of many of those species, because just like you and I, a rose species, genetically, it might be the same rose, but they can have variation in how they behave to diseases from one location to another. So we've tested multiple accessions as well. And that's some of our trial plots near Crossville on the Plateau Research and Education Center. Now, here is a picture of a large rugosa rose in front of a hospital in Murfreesboro, Tennessee, which is south of Nashville. And this rose looks great. Never have we been able to detect any symptoms, or when we test the rose, any virus titer. And we say, oh, we have a resistant one. Maybe, maybe not. You see, there were 2 beds of knockout roses, one on each side. The incidence of Rose Rosette before they took them out was 100%. So we say, well, we got it. But you know, in plant pathology, we've learned that there is a phenomenon called escapes. Perhaps the way the air currents were, because there were some burns and some trees and things like that. The wind currents just didn't blow mites into it. Maybe it's still susceptible. Maybe it's what we call an escape. It escapes the disease, not due to genetic resistance, but just due to the randomness of its location. And so what we have to do is take roses like that and test them in replicated trials where we have other roses that we know are susceptible. And if they get the disease and none of our replicates of the test rose get the disease, well, then we can start to say that, yes, it appears to be resistant. And to do that, we've had a screening program going on since 2015. And it was done at Delaware, Oklahoma, Tennessee, and Texas. And Dave Byrne, who is a rose breeder in Texas, was the leader on this particular project. But most of the roses tested were actually tested in Tennessee. Tom Evans tested some of them in Delaware, and Francisco tested, and Jennifer tested some of these in Oklahoma, but we tested them all. And then Brent Pimelton was looking at them at an experiment station for Texas A&M near Tyler, not for Rose Rosette, but to make sure if we found something that it wasn't a dog for other diseases. You know, what good would it be to put out a rose resistant to Rose Rosette if it was super susceptible to black spot? So, and then the people doing the genetic analysis, taking our symptomatic data and our virus titer data, was Dave and Oscar back at Texas A&M. So these 3 at the bottom are just examples to give you an idea of the types of roses we looked at. I told you over 1,000 cultivars. Many of the cultivars originated from the United States. We looked at a lot of Cortis roses and roses from England. So we had a bunch of roses that we tested from Europe. And then the Brenda Bella roses, which is a new group of roses that have been sold from a company in Florida, but they originate from Australia. And so we've looked at roses from different parts of the rose, hunting for resistance. And what we have found is about 3.8% of the roses that we have looked at, which isn't many, but it's still a significant number when you've looked at so many. We can't infect with the virus. They don't develop any symptoms. We can't detect the virus in them. And that's after we have been intentionally infesting them with viviparous mites multiple times a year for 5 years. So we feel pretty confident that these roses are immune. That means they can't get the virus at all. But then we found some more roses, not quite as many, that we label as resistant. They're symptom-free, or they have very minimal, almost undetectable symptoms, but they test positive for a low level of the virus. But the level of the virus over years never goes up. The symptoms never get more severe. Is this a good thing? We don't know, because what we don't know is that low level of virus in those plants still high enough that the mites can pick it up and move it to other plants. If that turns out to be true, these guys would be seen as typhoid Marys instead of being a benefit. So this is why research has to continue, because we need to determine if those plants are typhoid Marys or something useful. Now, we can take the ones that are immune, and be honest with you, none of those roses are anything you want in your garden. But we can take the genes of resistance out of them, and through breeding, we can breed them with roses that have highly desirable horticultural traits. And then we can test those seedlings, and then we can take the best of those and breed them back with that rose again. And now instead of being 50% highly desirable, they're 75%. Do it a third cycle, they're 88% desirable, but we test them, make sure they carry the resistant genes. Because we've been doing a lot of work with that. And this was questions about the studies. And so now where are we at? Well, you'll see in a minute that we have 2 resistant genes, and those resistant genes have been detected in both diploid and tetraploid populations of roses. And this is really, really good news. And so I have this year 1,200 seedlings coming for 3 years of evaluation. And we'll be planting them in Crossville in May, because our last frost date in Crossville up on the Cumberland Plateau is May 10th. So in about the middle of May, when that last frost date has passed, we'll be putting these seedlings out, because they're too valuable. Many of them are likely to be carrying Rose Rosette resistant genes, and also be carrying genes for desirable horticultural traits. So these seedlings are literally worth their weights in gold. And so we'll be planting them out, and we'll be testing them. But when we test them, that means phenotyping. So what we do is we plant them out, we evaluate them, we collect data for 3 years. Twice a year, the plants are assayed for virus. You know, a virus assay, a nested PCR assay. That means you have to run PCR twice. Not the labor, just the reagents and the chemicals and the primers that are involved. Is $8.67 per rose. And we have to test that rose twice. And I have 1,200 roses showing up. You get an idea how expensive that is? That's really, really expensive. But we know where these genes are. One is on chromosome 5, and we know where it's at. The other one is on chromosome 1. And so we can test to make sure the roses are resistant, and that they don't carry virus. And then we can also confirm through marker selected breeding that they have the marker for, they have the gene for resistance. Then we got something. And we're just a couple of cycles away for having roses for your garden that have resistance to Rose Rosette. You know, 5 years ago, we didn't even know. We thought it was a crapshoot, looking for a pin in a gigantic haystack. And, we didn't think we had much of a chance, but now we got it. And so now we just got to get it into something that we can use. So this is what a field looks like when we put these little plants out. And you can see how tiny they are. And then in the third picture down where the guys have been over, they're actually infesting every plant with 800 to 1,000 viviparous mites. And we do that twice a year. And in addition to that, we have rows of sick plants spewing mites. This is not a place you want to be if you are a rose, because we're doing everything we can think of possible to get those roses infected with Rose Rosette. We don't want escapes. We want to make sure that if you survived in this field for 3 to 5 years, you are rock solid for resistance. And we're going to test them. We test them all to make sure, okay, you're not showing symptoms, but you got the virus. We don't want those guys either. They might be the typhoid Marys. So this is what we have to do. It's hard work. It's labor intensive work. And unfortunately for me, it's very expensive work as well. So this comes down to, well, what can we do? We're applying for grants. Last year, our grant was rated very high for funding. But you know, they had over 200 excellently written proposals and they only funded 5. We were in the top 15, but we didn't get funded because they only funded 5. And they didn't fund a single ornamental proposal last year either. Now we'll be trying again this year. If we don't get the money, this research is going to stop. It absolutely will stop, and we'll just walk away with unfinished business, and we won't have roses for you. Now, Mr. Paul Zimmerman, who has Paul Zimmerman Rose Gardening on Facebook, has put together some videos that you can watch, and he has a GoFundMe page. Now, his GoFundMe page is audited by Mr. Chris Van Cleve. Some of you know Chris. He ran for president of the American Rose Society a few years ago. He's a senior vice president for Regions Bank. And Chris has agreed to audit this account every 3 months for free. And so he does a complete audit to make sure everything's on the above board. We've already received money this way. You know, any donation, we've had donations as much as $5,000. We've had donations as low as $4. You know, this neat thing about money, it all adds up. It all adds up. Now, if you want to make a donation directly to the Rose Rosette research at the University of Tennessee, and Kimberly, this is the information I'm going to be sending you. And you want to send a check. You send a check to Mr. Tom Looney, Director of Development, and there's his address. If you have a question, I've got his phone number and cell phone number listed, and I'm going to send all this information to Kimberly. And there's his email, or at the UT Foundation, or you can email me and I can send you the information as well. And all the money will go to, right now, we think we have money to put the roses in the field. Any money, more money that's raised goes towards rose testing. Because, you know, $8 for each of those 1,200 seedlings, plus another 200 that we have from some work from the year before. We have a and you do that times two, both in June and then again in October. It's a very expensive proposition. And I want to acknowledge some places as well that have already given Rose support. The Heritage Rose Foundation has been very supportive of us. The American Rose Society Research Trust has been too. The American Garden Rose Selections has. The Rose Council is paying to have all the roses put in, both in Texas or in Oklahoma and in Tennessee this summer. And then there's private donors like you. Now, I talked to Kimberly just a few minutes before we started this presentation, and I'm looking very excited because the next time I give this presentation, I suspect that there will be a 6 line on here, and number 5 will become number 6, and and what will take its place as number 5 will be Rose Geeks.
Yep.
And so because y'all have done a wonderful fundraising thing with your calendar, and we're very appreciative. Everybody in the Rose community knows about what y'all are doing, and we can't thank you enough for your support. But it's people like you that are going to make the difference as to whether or not we succeed in developing roses resistant to Rose Rosette, so that all those other things I talk about you having to do and watch for will just go by the wayside. You see, all those management strategies I gave you, we look at those as short-time solutions. Resistance is a long-term solution, and that is what we have to do. But so we're going to get there. And when we get there, as we learn more about Rose Rosette, as we're able to produce roses with resistance to rosette, now that we have it, but the roses also have the horticultural characteristics that you want, whether it's a hybrid tea, a floribunda, a grandifoliar, shrub rose, mini flora, miniature roses, a climber, regardless, and colors, of course. And I don't know about you, but a rose to me personally is not worth a dang if it doesn't have fragrance. So you see, but this is the kind of work that we have in front of us, but we're going to get there. So one day, we're not going to fear Rose Rosette, but there's still some things we're going to be afraid of. Alligators walking through nurseries where roses are being grown, that's something that I'll be afraid of for the rest of my life. And the bottom picture, a good friend of mine, Wayne Myers, who's been a rosarian for a long time in the Jacksonville, St. Augustine area. He used to live more in Orange Park across the St. Johns River from Jacksonville, but now he and Lindy live in St. Augustine. He was weeding his garden for the first time in 2019, and he was on his hands and knees pulling weeds, and he came nose to nose. It was a little cold. The ground was cold. So fortunately for him, that Eastern Diamondback rattlesnake was cold. So it just sat there coiled looking at it. And they were just inches apart, nose to nose. Now, I don't know about you. Rose Rosette we're going to beat, but the rattlesnakes are still going to be there. So alligators, rattlesnakes, I can't help you with that. But we will defeat Rose Rosette, especially with your help.
I was curious, when we talked about gall and we talked about sanitizing everything, when you're working with a rose that you are pruning out the mites or a shovel, do we need to be concerned about sanitizing?
No, you see the difference is the mites can't survive in the soil. The bacterium that causes crown gall can.
Got it.
So, you hang your shovel up, the mites ain't gonna survive on it. Probably on that, it won't survive a couple of days. Now, even if you use the shovel right away in another bed and you introduce some mites to the soil, they can't feed on the roots.
I see.
And they're going to die.
Got it.
And they're tiny. They're not gonna be able to crawl up. They can't get a hold on a dirt clod in a balloon. So there's no need for that kind of stuff. This virus cannot be spread mechanically on pruners either. like the crown gall bacterium, agrobacterium tumefaciens can. So no, we don't have to worry about those kind of things with the virus, and thank goodness.
I really like hearing you talk about it because you demystify a lot of the, concerns about Rose Rosette Disease. It is very scary and it needs to be solved, but I really appreciated hearing you say that if you can't get to it right then, just take down the population really quick, cut out the branch that's
Cut out the rose.
affected.
But you see, we didn't know that a few years ago. It was scary. 2014, when I first got into that, all we had were black boxes. We had a meeting of people that were scientists that many of them decided not to, but might be interested in doing research on Rose Rosette. And that was a meeting put together by Star Rose, when Steve Hutton owned and was president of Star Roses. And so he flew us all up to Pennsylvania for a meeting. I think actually the meeting was in Delaware, where a hotel was close by, but right across the Pennsylvania line. But the bottom line is, we put up what we knew about Rose Rosette. We knew it was killing millions of roses, but we knew so little. So practically everything I told you about Rose Rosette, we have learned since 2015.
Amazing. It's great work.
So, but it doesn't end, but we'll continue. Time is not on our side. I'm retiring July 1st.
Of this year.
Dave Byrne is already 65% retired. But in retirement, I've agreed to continue doing Rose Rosette resistance research. All the other will go by the wayside with me. And we'll do it for 3 years. And so we'll get this next round done before I'm gone. But you see, I'm not the only one. Ornamental pathologists are all about the same age, and we're retiring. We will not be replaced. There will not be a replacement for me. There will not be a replacement for many of my colleagues in other states. When I came to Tennessee in 1985 as a new assistant professor, there were 17 ornamental pathologists in Tennessee and in the states around Tennessee. When I retire July 1st, there will be 3. And one of those will be retiring in 2 years and another one in 4. And why aren't we being replaced? Well, you see how we're struggling to get money. They're interested in hiring people who can bring in lots of money. A young man in animal science just got he's only been here 3 years, and he just got his 5th multi-million-dollar grant proposal in working with chickens. This one is on coming up with new ways to keep chickens happy. That's what the title was. How do we keep our chickens happy? And, you're sitting there going, okay, well, the deans see this. So they want more chicken happy scientists and fewer ornamental pathologists, because the chicken happy scientists bring in lots of money. So things are getting skewed in that direction, and we don't seem to be able to stop it, and so, I hate it. That's why I'm hanging on for 3 years to do that. If I knew they were going to hire a young man or a young woman to take my place, I wouldn't have to. But that's not going to happen. And I love roses too much. Too many people have made an investment in this. We can't walk away till we get the job done.
Well, I'm glad that you're giving us 3 more years. And so that is the target that we'll all be driving to figure out how we can keep supporting you. And so I've got the Rose Geek calendar that we've been able to put together in 1 month. We were able to.
Yeah, it's phenomenal.
Thank you. So we'll be writing a check this week for nearly $1,000 after 1 week. And so anybody who's watching this video, I hope you are as inspired as I am and all of the other Rose Geeks. Please consider giving to either the GoFundMe. I have a couple of calendars left. You can reach out to me and I will get one to you. Look for our next calendar. We're going to be doing this yearly, and hopefully it will be even more that we can get in the coming year. Or please give directly to the address that Dr. Mark provided.
Or just contact me,
Yeah.
mwindham@utk.edu, and I'll get the information to you.
Perfect. And also, anybody who wants the PDFs, Dr. Mark sent those to me. They're on the Rose Geek site in the announcements. You can go ahead and pull those down. And then also in that calendar that we put together, we have a beautiful PDF that shows you Rose Rosette. Thank you, Dr. Mark.
Thank you so much for having me.